So let's move on to natural selection. And you already know what natural selection is, right? It refers to how organisms survive because of their adaptations, which give them an advantage in specific habitats. Well, Darwin's idea of natural selection came under many different assumptions and inferences. The first was that certain phenotypes give advantages in certain habitats. For example, differences in pigmentation can mean the difference between life and death in specific habitats, right? Let's say, pigmentation that helps with camouflage can help increase chances of survival, but that pigmentation has to match the color of the specific habitat that you're looking at. Because if you put that organism in another habitat with the pigmentation sticking out, then it won't really help. Okay? So the second was that in a population, certain individuals will possess some of these favorable phenotypes. So this means that these individuals enjoy much greater success in surviving and reproducing. Okay? By the way, we're talking about the individuals that possess the favorable phenotypes. Okay? So what does this show? This shows that there's a difference in individual fitness levels within a population, right? The higher fitness levels can be seen in the individuals with the favorable phenotypes. Of course, these individuals with higher fitness eventually produce offspring that also possess the advantageous traits that they possess. And these offspring are produced in numbers that are more abundant, or usually produced in numbers that are more abundant, excuse me, than what the environment can support. So remember the concept of K here, okay? Which is, excuse me again, carrying capacity. So, with these taken into account, to be technical about it, the process of natural selection results from increased or decreased chances of survival and reproduction because of the phenotypes possessed by organisms. So decreased chances of survival and reproduction if there are no phenotypes that can be considered adaptations. Thus, no advantages are gained, right? On the other hand, with adaptations, increased chances of survival are enjoyed by the organisms that possess the favorable phenotypes. Of course, again, this operates on the basis of these traits being heritable. If they're not heritable, then the advantages can't be passed on to offspring. That's not really natural selection. So the first point we made earlier was that there are genetic variations within a population, right? That these variations can lead to some individuals being better suited for the environment with a higher fitness. Well, how do these variations come to be? Well, variations in a population come from both a genetic and an environmental basis. I'll give you an example. So in the 1940s, a study by Jens Clausen, David Keck, and William Hisey studied or focused on a population of Potentiella glandulosa, which is a plant. So they took P. glandulosa samples from different environments, so some from upland, some from lower elevations, and they cloned them. And then they further planted the clones in different environments and observed the differences that they showed after growing. You can see the differences in the picture here. Of course, they found out that these different environments produced highly varied plants. One environment produced smaller plants, another produced larger ones. One environment even had plants that produced more flowers. Okay, so what does this show? Well, this shows that there are still genetic variations within populations of the same species. Okay, in this case, plants from the plants from the lowlands have subtle differences from those from the highlands, right? So yeah, same species but with small and subtle differences. By the way, these differences are known as ecotypes. Okay? Ecotypes are populations of the same species that are genetically distinct from each other. But since they're still of the same species, they can still interbreed. Okay? To help you understand this better. By the way, so the alpine plants, mid-level elevation plants, and lowland plants, these are all ecotypes. They're of the same species, but they're still genetically distinct. Okay, but yeah, again, to help you understand ecotypes, if you're again familiar with Pokemon, so we're using Pokemon examples a lot. Imagine the regional variants to be ecotypes, right? So we have a Vulpix from one region being fire type, and a Vulpix from another region being ice type. They're the same species, but they still have genetic differences, and they can still interbreed, okay, since they're still both Vulpix. So, what's the importance of studying these, studying ecotypes? Well, in terms of conservation, it's very significant. If you know which populations are distinct from each other, then you know which ones to protect, right? Well, we'll discuss this in more detail later on. So always remember, heritable traits must have a genetic component. So gene frequencies are dependent on the heritability of traits. This is because the changes that happen to one generation must persist in the population, right? As such, evolution cannot happen without heritable traits. Of course, like other biological concepts, there's a model or an equation that helps identify the factors that can cause changes to gene frequencies in a population. Okay? So in this case, this is known as the Hardy-Weinberg equilibrium model. Some people pronounce it Weinberg, but let's just pronounce it Weinberg. So we mentioned before that evolution technically is the change of gene frequencies within a population in question, right? Well, the assumption in the Hardy-Weinberg model is that the genetic variation in a population remains constant if there are no factors that disturb it. Thus, the term equilibrium, right? No disruptions. So since there are no changes that occur, you can actually view this model as a null hypothesis, in that it supposes that no changes happen from one generation to the next. So, for no changes to happen between one generation to another, specific conditions must be met, which are listed here. Okay, let's go through them one by one. So the first is random mating. So, males and females just randomly choose mates, which means that no sexual selection occurs, which doesn't really happen that much in nature, right? Next, we have no mutations. Why? Because mutations in genes can introduce or change alleles. Thus, mutations can disrupt the equilibrium. The third is large population of individuals. This is because large populations are more resistant to genetic drift. Why is this important? Remember that genetic drift refers to changes due to random chance, right? Well, smaller populations have a higher chance of losing alleles or reducing the frequency of alleles due to random chance. Thus, large populations are required for the Hardy-Weinberg equilibrium model. The fourth is that there's supposed to be an absence of migration. This is pretty obvious. Either immigration or emigration can cause alleles to be lost or gained in a population. So that happens when individuals move, right? And finally, we have equal survival and reproductive rate for all individuals, which means there's an equal fitness. So with equal fitness, individuals have an equal chance of surviving and reproducing. This means that there are relatively no changes that can occur in the alleles. Why? Because if fitness levels are not equal, some individuals will enjoy greater reproductive success, meaning that they produce more of their offspring, meaning that their offspring will inherit their specific phenotypic variants. Again, this goes against the suppositions of the Hardy-Weinberg equilibrium. So basing it on these 5 conditions, you'll notice that these almost never happen in nature all at once, right? So, if these never happen all at once, what's the point of the Hardy-Weinberg equilibrium model if the conditions are never met? Well, remember that this can be considered a null hypothesis. This means that it gives scientists a way to compare a specific environment or population with a null hypothesis and gauge its capacity for evolution to occur. The higher the deviation away from the Hardy-Weinberg model, the higher the chances for changes to happen, right? So you basically go against the null hypothesis in this case for changes to happen. So this equation here is an expression of the Hardy-Weinberg equilibrium model. How can it be used? So we mentioned that the model is highly dependent on looking at gene frequencies, right? Well, the equation can be used for calculating gene frequencies, okay? So we have this equation, p plus q squared equals p squared plus 2pq plus q squared, or is equal to 1. At the same time, I want you to look at these genotypes and phenotypes here, which we will use to explain the equation. Okay, so we have a population of lady beetles with the genotypes SS, AA, and SA. Now, focus on p squared plus 2pq plus q squared equals 1. Okay, focus on this part. P squared refers to the frequency of the SS genotype, which is this one. Okay, 2pq refers to the frequency of the SA genotype, whereas q squared refers to AA. Okay, so we have AA here and SA here, and all equal to 1. So in short, we just take into account the mixes of the genotypes in the population, right? We just take into account the mixes of SS, AA, and SA in the population. So suppose that the frequency of beetles possessing the SS genotype is 81%, or 0.81. SA is 18%, or 0.18, and AA is 1%, or 0.01. What you then have to do is you calculate for the frequency of the S allele and the A allele. How do you do so? Well, you use the equations shown here. Okay, so if you use those equations, you can get the frequency of SS plus 1/2 frequency of SA to get S, right? And you can use frequency of AA plus 1/2 the frequency of SA to get A. Okay, so what are the frequencies then? So for SS, it's 81% or 0.81 plus half the frequency of SA, which is half of 18%, which is 0.09. So you get 0.9 for the frequency of S. Okay, that's for the frequency of S alleles. On the other hand, for A, it's 0.1. Okay? So again, take note of that. S is 0.9, A is 0.1. By the way, what do these numbers mean? So we have 0.9 and 0.1, right? So this is 90% and this is 10%. Okay? So this means that 90% of eggs and sperm carry the S allele, and 10% of eggs and sperm carry the A allele. Now, again, the Hardy-Weinberg equilibrium model assumes random mating, right? So since the assumption is random mating, we can predict the results of mating since we have the percentages for the S allele and the A allele, right? Because you know that 90% of them carry S, that 10% of them carry A. So you can actually predict the results of the next generation since it's a random chance of mating. Okay? So how do you do that? You just look at all the permutations. Okay? All the permutations between S and A, between sperm and eggs. Okay? And in this case, we have S from sperm, S from egg, S from sperm, A from egg, A from sperm, S from egg, and A from sperm, A from egg. What does this mean? S from sperm fertilizing an egg that contains S. Okay? S from sperm fertilizing an egg that contains the A allele. and so on. So how do you do that? You just multiply the values. Okay, so we have 90% and 10%, right? So again, S times S, S from sperm, S from egg, so it's 90% times 90%, which results in 0.81 or 81%. Okay, S from sperm and A from egg, so it's 0.9 times 0.01, getting you 9%. Okay, and so on. So this is the spread. SS is 81%, SA is 9%, AS, also SA, is 9%, and AA is 1%. So going back to the equation that I showed you earlier, this one, okay, we just plug everything in. Okay, so again, P2 is SS, 2PQ is SA, Q squared is S or AA. So you just plug in the values and then you get the frequencies, right? So it's 0.81 plus 0.18. Why is it 0.18? Because we have 2 of 0.09, right? So you add those two. One from SA and the other from AS, which is also SA. So plus the final one, which is 0.01 for the 1%. All equal to 1. And that's what's shown here. So yeah, what's this for again? Well, again, this is just a model. Okay, this predicts the frequencies of alleles within a population, as well as the hypothetical chances by which these alleles and their combinations arise. So how specific certain phenotypes are selected for in natural selection can be classified into 3 types. Okay, we have stabilizing selection, we have directional selection, and we have disruptive selection. The main difference between these 3 lies in the quality of the phenotype. Think of it this way. See that you have the trait body size. You can gauge whether the type of selection is directional, disruptive, or stabilizing based on the quality of the phenotype after natural selection has selected